Okay, so what I want to do today is just talk to you a little bit about my work on islands. And so, yes, as Jose Maria said, I'm going to talk about adaptive radiation, in particular, focusing on Hawaii, on the Hawaiian Islands, and talk about the nuances behind adaptive radiation. So, I was thinking about this over the last few weeks, just thinking about this. I mean, that it was a huge honor to be talking about to be doing the Alfred Russel Wallace Award, and or talk. And so, what in doing this, I thought, well, what honors Alfred Russel Wallace? And so, one of the things I was thinking about was that he was such a naturalist. And this is what I want to emphasize kind of from the get-go, that Alfred Russel Wallace, in the words of Sandy Echternach, was a consummate natural historian and evolutionary biologist. And so I want to emphasize from the beginning just how I became involved in the natural history and understanding of islands through the natural history context. So first of all, how did I get to be working on islands? As Jose Maria said, it started with, for me, in Scotland. I come from the southwest of Scotland, where it's cold, sometimes snowy, mostly drizzly, wet. But I came over to the US to do my PhD, and there I worked with Susan Reichert, who is a behavioral ecologist. And so I was interested in understanding the behavioral ecology of spiders. And I'll come back to spiders very shortly, but spiders are very good for understanding details about ecology and behavior, because they do both just while you're sitting watching them. So I studied this spider, Tetragnatha elongata, that tends to live over water, any wet place. It's very common on the east coast of the US where I was studying. It's very common actually just about everywhere. And they all look very similar, with long bodies, long legs, and anyway, they were really nice to work on. They were very, very straightforward. And this is important background, and you'll see in a second. So then I came over to Hawaii. I came over to Hawaii to do behavioral ecology on the Hawaiian happy face spider, which is a fascinating animal in itself, but I'm not going to dwell on it very much because it's kind of another story. But they're very colorful animals. They've got this balanced color polymorphism. But as I was working in Hawaii, I became aware of another group of spiders. So first of all, just a little bit, as most of you know, Hawaii is extremely isolated. It's right out in the middle of the Pacific, the so-called most isolated archipelago in the world. And when you think about Hawaii, at least when I was thinking about Hawaii, what was known about Hawaii, my understanding was Hawaii is just a kind of idyllic paradise with lots of beaches. And as Mark Twain said, the loveliest fleet of islands that lies anchored in any ocean. And so that was kind of my image of the Hawaiian Islands. But what happened when I got to Hawaii was that I started looking first for happy face spiders in high elevation habitats. And these are very different. They're in when you go up in elevation in Hawaii, the endemism is round about 99%, at least for insects and spiders. And so you're kind of putting your eyes upward in the islands and going into this forest, which is extraordinarily lush, wet, and often quite cold. And when I got to Hawaii, way back in the dark ages, I got there in 1987. And the knowledge of biology largely rested with this person, R.C.L. Perkins. R.C.L. Perkins was sent out from from the British Museum. He was based in England, but came over to Hawaii to understand the fauna. He wrote the Fauna Hawaiiensis. So he worked in the islands from 1892, resulting in this extraordinary tome, the Fauna Hawaiiensis, in 3 volumes. And he was an extraordinarily insightful man. He, one of the, a few, just one quote, that species arose through a series of events involving divergence and geographical isolation, followed by speciation, with competition increasing as new species were added. I mean, imagine, this is, the late 1800s. And this is, Darwin's theory was just coming about and was not widely accepted. And you have Perkins just sitting out in the islands making all of these observations and just learning about the biota. And here again, what I want to emphasize is his very deep understanding of the natural history of the biota of these islands, kind of following in the footsteps of both Darwin and Wallace to really know the organisms he was looking at. And so he conducted many years of field-based research on multiple groups and got extraordinarily insightful ecological observations. So this is where I come in. What about the spiders? So when I got to Hawaii, the knowledge of the spiders was really, it was based on the work of R.C.L. Perkins. And so when I reached the islands, what was super exciting was that there was Tetragnatha there. Now, just let's think about how the Tetragnatha that I'd done for my PhD. Remember, I said they were boring, they were over water, they're very not boring. They look very uniform, and all with long bodies, long legs, stretch out their legs. They're sometimes called stretch spiders. Anyway, what happens in Hawaii? First of all, how did they get there? And so when you think about how spiders get to these places, they do have a capacity to balloon. This is a behavior that can be selected on, so they can do it, and then selection can act to reduce that ability. But what they do is they let out silk. You see the wind catches it. You see here it's letting out silk. The wind pulls, and then boom, off it goes in the air. You see the wind here pulling, and then boom, off they go in the air. And so this is how spiders disperse, and this is how they can get extensive distances. So when you look at Hawaii, then you look at high elevation habitats. What has happened there? The Tetragnatha spiders have reached those islands. They've diversified in there. And what's more, as I was saying before, what makes spiders really cool is that you can really watch their behaviors. They tell you basically where they want to build their webs, how they want to build their webs. You can look at exactly what they're doing, where they're doing. As you see here, this is one's eating something. You can say, oh, what do they like to eat? And when do they like to eat it? And so they tell you everything about their natural history and how they differ. And so they're very straightforward to watch in many ways, and it's just understand the ecological context within which they evolved. So long-jawed spiders in Hawaii, they're just super diverse. So remember these long skinny things? No longer. You've got all of these different species that are confined to different places. Some of them, this one here is confined to East Maui. This is the summit of Oahu. There's an entire lineage that is abandoned web building and just runs around as cursorial predators running around the vegetation. This one is confined to the summits of Oahu as well, and just seems to feed on amphipods. They're all extremely specialized and extraordinarily diverse. So the question is, how did they diversify? And this is kind of the holy grail of adaptive radiations. understanding the mechanisms of diversification. So this is where I've been thinking about this for a very long time. And so a few years ago, as Jose Maria said, I was the president of the American Genetics Association. So the plan here for this, we organized a meeting, and the plan was to try and figure out, are there generalities in that underlie adaptive radiation? So what I did was get a whole bunch of people together who study adaptive radiations and try and figure out what the commonalities were. So there were people looking at Galapagos finches, Andrew Henry, for example, Hawaiian honey creepers, Rob Fleischer was there, Anolis lizards, James Stroud was there, stickleback, cichlid fish, Ollie Seehausen, and pupfish, Chris Martin was there. Everyone gathered together, Kerry Shaw for La Pala crickets, Jim Mallet for Heliconius. We got all of these people together to try and see what the commonalities were that underlie adaptive radiation. And this is what I want to focus on. So in doing this, we wrote up a kind of overview that highlights really what adaptive radiation means for all of these different radiations. And the bottom line is that it means very different things. And this is what I want to emphasize. So first of all, what are the different things when I say, it means different things? So first of all, we talk about one nice example is Acatinella. This is a radiation of snails, in particular on the island of Oahu, extremely diverse. And many of them are extinct, so huge conservation concerns. What's interesting about these snails is that they're microallopatric. Each one occupies a slightly different area. So radiations can occur by just really doing the same thing, but occupying a different area. And this is true, some birds have diversified in the Solomon Islands in the same way. Salamanders in the mountains of California have diversified in allopatry in the same kind of environment. But together, they form a radiation. So this is one, it's sometimes called a non-adaptive radiation, although that term is kind of, doesn't, they're still adapting. But the thing is they're diversifying within the same kind of habitat, but different geographic areas, sometimes tiny little areas where they diversify. So that, sorry, that's one way of diversifying. Another is one that we're quite familiar with, in particular through the work of Patrick Nozel and Dolph Schluter, who wrote this incredibly impactful book on adaptive radiation. But the idea is kind of based on fish, in particular stickleback fish, where they change ecologically in parapatry or allopatry. So you get the formation of kind of ecotypes. So they go into different environments. And then you get, when they come into juxtaposition or they come into close proximity, you can get reinforcement. This was sometimes called the Wallace effect after Alfred Russel Wallace. The idea is that the hybrids don't function well in either environment. So when they come together, there's selection not to have hybrids. So this is a very clear example of ecological speciation, divergence into different environments and selection against the hybrids in the areas of overlap. However, there are other ways of diversifying. And in certain contexts, you can get the initial divergence in the same environment, often in allopatry. Then when taxa, sibling taxa, sister taxa come back together, they're basically the same thing. They're in the same environment. They're going to compete madly in terms of, in particular, natural selection. And so you get divergence within the same environment, divergence in niche space. And this is where you often get, like in the anolis lizards, you get formation of ecomorphs. It's ecological and reproductive character displacement in the secondary sympatry. The divergence is caused by the interaction between these sister taxa. Okay, so let's come back to Hawaii and see what you find here. So thinking back to Hawaii, what makes Hawaii particularly special is that it's a chronosequence. So what does that mean? It means that the lava emerges, it's still doing it very actively. It emerges in the kind of south part of the archipelago. The lava comes up, forms volcanoes, which can be massive, like Mauna Kea, Mauna Loa. But over time, these volcanoes that are formed move off on the Pacific plate. So as a result, you get very young lavas, very young landscapes. And then over time, you get older and older and older environments. And so that means you've got the formation of communities across this sequence, and their temporal arrangements. So the oldest communities on Kauai, the youngest on the Big Island. Now, is that true? Do you really get organisms, the oldest groups on the oldest island, the youngest on the youngest? What you find in many groups, and this is something that Warren Wagner and Vicky Funk initially drew attention to, the idea of progression, or the progression rule. So the idea here is that in most groups, or many groups at least, the colonization started on the oldest island, or even before, sometimes later. But when it started on Kauai, those groups that did start on Kauai, here I'm showing it through Carrie Shaw's work with Laupala crickets. So they start on Kauai, and then you see two lineages are formed. One hops down, here's Kauai, and then hops down Oahu, Maui, the Big Island. The second lineage, Kauai, and then hops down Oahu, Maui, and the Big Island. So why do they do this? It seems that the first ones that establish new populations on a new island, they monopolize their resources, they adapt, and that blocks subsequent colonists from establishing. And so this, because of this temporal arrangement, you can really get insights into the process of diversification. Okay, so let's come back then to the spiders and see what's going on here. Do they show progression? That's the first thing. So do they jump from the oldest island to the youngest? The answer is yes, they do, but they do so in multiple lineages. Different lineages are hopping down the chain, and this is super important. So you get progression, but separately. So what we need to do then is look at different clades and ask ourselves, what is going on in these different clades? What is the mechanism of adaptive radiation in these different clades? Okay, so first, we'll just pick off one group here, and I want to show here a group that has undergone what I would characterize as a non-adaptive radiation. So this lineage has basically gone up, it has initially adapted to dry environments, and it's at low elevation and high elevation. But what it's done is it's hopped from the oldest island to the youngest island, but always in these very dry environments, mostly above the inversion layer. So it's dry, open habitat, and they stick to that. They don't do much else. They just hop down the island chain. Kind of a classic example of niche conservation. One lineage is adapted to these environments and then just hop down the chain. Okay, so you say, well, that's a fairly straightforward story. They're mostly, they're entirely allopatric, and they just occupy these dry environments. But is that the only story? So let's look at other stories. Now, here's another one that I think is just really it's just very nice. It's the story of these Oahu elongate species. That these ones actually look a bit like mainland Tetragnatha, but what they have done is they've adapted, but they conserve their habitat. Their primary axis of differentiation involves conservation of the habitat. So relatives occur at high elevation in these very wet environments on Oahu. Relatives occur at lower elevation on Oahu. So basically what's happening is you get relatives, so related species on the summits of the mountain and then relatives at lower elevations. So again, niche conservation, allopatric divergence between similar forest types. Now you get this kind of thing in other spiders. I'll just mention here the one other group of spiders in Hawaii that's not Tetragnatha. So these ones, these are Mechaphisa crab spiders. And this is the work of Jessica Garb, who was a graduate student in my lab a while back. So Jessica studied the adaptive radiation of these Hawaiian Mechaphisa. Very diverse. What I want to show here, this is the dark blue is Kauai. And what you see here is on Kauai, they have diversified into almost every kind of niche available. They occupy all of these different kind of arenas in an environment. What's interesting is that they've done that on Kauai. And what seems to have happened is that they've then simply hopped down the island chain. So you go from Kauai and every lineage hopped down to Oahu, to Maui, to the Big Island. So it's just this, again, a kind of non-adaptive radiation within each of the lineages with early ecological diversification on the oldest island. These ecologically similar entities then diverge in allopatry. And perhaps the best example of kind of non-adaptive diversification is in this sheet web spider, Orson Welles. This is the work of Gustavo Hormiga, a colleague at George Washington University, and Mikel Arnedo. And he was interested in how these things are diversified because you get them on all islands and they do just basically the same thing. Huge sheet webs. And you know, they're everywhere you go. So he did sequence analysis. This was a while back now, 2003. And what he showed was that they've diversified on all islands, and they show the classic island hopping from the older to the younger islands. And you get 5 species on Kauai that have differentiated into each different habitat, geographically separated in that island. 3 on Oahu, 3 on Maui Nui, and 1 on the Big Island. So what's basically happened, and I should say similar things go on with Laupala crickets, again, the work of Kerry Shaw, divergence largely in allopatry and the so-called non-adaptive radiation. So let's then, we've got this kind of story of non-adaptive radiation here. Is that what's going on with all of them? And so I'll just look at some other groups here. And so here's one I want to show you. This is a really beautiful example of ecological speciation. So ecological speciation, this is one I think we're very familiar with. And to use the definition of Patrick Nosal, the process by which new species form as a consequence of divergent natural selection between contrasting ecological environments. So where has it happened in these long-drawn spiders? Well, you see these 2 lineages on this one is on Maui. This one is mostly on Hawaii. Well, what we're showing here is on the Big Island. What you see is that they've diverged between similar habitats, going from dark lava type habitats like you see here. So this one you find on the dark lava habitats. This one here, the white one, you find in dry forest on this Usnea lichen. So there are different environments, different substrates, and it seems that selection has pulled them apart into these different environments. So you get divergence between sisters much greater on Maui, presumably because there's been more time. And so what's going on here is basically selection, presumably ecological speciation, is pulling these things apart and into these very different environments. They're largely, you see here's the map to show where they are on the Big Island. They're adjacent habitats, but they look totally different, and the environment is just selecting them in different directions. And again, you get this kind of ecological speciation just beautifully illustrated in other systems in Hawaii. I think silver swords are just a very nice example where you get different substrates or different environments associated with different species. Selection is pulling these different species in different directions. And so you get, you know, that high elevation species here. You get some that are on Pahoehoe lava, some on A'a lava. They're different substrates, close proximity, but selection is pulling them apart. Thinking about spiders, our one sojourn outside Hawaii. This is Frederick Henrich's work with his student Charlotte de Bashir. So he was looking at wolf spiders in the islands of the Galapagos. In wolf spiders, you find this same kind of thing. They've adapted to high elevation, low elevation. On each island, they go high and low. And actually, Kaina Privet, who's a student with me, and Julian Petillon in Rennes, she's looking at the similar kind of thing in wolf spiders in Hawaii. But it seems that it's the same thing. Selection is pulling them into different environments. Classic ecological speciation. Okay, so that it kind of you say, well, is that the end of the story? And no, it's not. So let's look and see what happens in other lineages. And here I want to just focus in on a bit on a couple of lineages because I've done the most work on these particular taxa. So here, I'm going to look at this one first. This is a species that, or rather a lineage that you find on Maui Nui, so the islands of Maui, Molokai, Lanai. But I'm going to focus mostly on East Maui, the forest there. So what has happened here is that you get a lot of close relatives that are living together, and what you find is that they've shifted their niche within an environment. So first of all, let's look and see what I mean by within an environment. And this is a work of Anna Hiller, who was an undergrad student in my lab. And this is just looking at niche modeling. She was trying to see how sister species in these different radiations of flies and crickets and plant hoppers, how sister species differed in terms of the kind of environment in which they lived. And what you see here, the spiders, the Tetragnatha spiders and the Drosophila flies, what she found was really that their environment is stuck in this very small range of niche space, environmental niche space. So this is rain, vegetation, rain temperature. And so they're just diversifying within the same kind of forest. And so let's just think then about, you get all of these sister species living together in the same forest. So what's going on here? First of all, I'm looking at, these are web builders, and this is the work of Susan Kennedy, who is a student in my lab. And Susan looked at the niches occupied by these sister taxa. Are they really different? Well, they sure look different, This one is, kind of long and brown and dull, and this one is bright green and bumpy, and this one is silvery orange. Silvery kind of maroon. And they spin different webs. She looked at the niche properties that in these different species, and so looked at isotopic signatures, web architecture, site choice, gut content, and you can see that these are the different species, and you can see marked difference. This is the nitrogen isotope, this is the carbon isotope. The nitrogen is the one you'd expect to be more divergent within the habitat, and it certainly is. Web architecture is very different. The height that they build, the angle that they build, and the gut content is, all of these suggest that these things are occupying fundamentally different niches within that environment. So these are all web building tetragnatha. So what exactly, let's just look in particular at the web architecture. What you find in that habitat, and this is the work of Todd Blackledge, who was in my lab, but Todd looked, this is on Maui, where Susan was looking, and you see that the web architecture within that area is just totally different. So this is one species with an open web. This is a big closed web with this species. This is a tiny little web that's built under ferns. So very different kinds of webs. They have evolved, similar web types on other islands, but they've diverged within a site so that these species can presumably occupy different niches within that environment. Okay, so let's see what's going on here. You get convergence within, this is Oahu. Here you see the different web types. This is Maui at the bottom. What's interesting here, you get similar web types here, for example. This is Oahu, this is Maui. And you say, oh, they show ecomorphs. And well, do they? Are the spiders really that similar? Oh no, they're not at all similar. So the web forms have converged, but the spiders themselves have not at all. So you get convergence along multiple different kind of niche axes, so to speak. So now let's look at a group that I know even better. And so I can really talk in detail about this one a little bit. These are the spiny-leg tetragnatha, and they have diversified. These ones do not build webs, and they've diversified across the wet forest on all islands. And what's cool about them is that they show repeated evolution of the same form in the same way as the Anolis lizard. So basically what you see, this one is Kauai with a big brown ecomorph shown in dark brown, green, and then the light brown shows the little brown ecomorph. And you get on Oahu is the appearance of the maroon ecomorph, green, big brown, little brown. And again on Maui, maroon, green, little brown. And then there's other things going on, of course, but basically the important thing is you get repeated evolution of the same form on Kauai, Oahu, and Maui. And then on the Big Island, it's just populations from the older islands. So how do you get this? How do you get the formation of these ecomorphs? So what's nice is we've got this time frame to look at this. So let's, if we look at how the ecomorphs have formed. This is Kauai, this is Maui, and you see this is the green ecomorph. They look identical, but they're actually independently evolved. This is maroon on the different islands, again, independently evolved, big brown, and here you've got little brown. So a student in my lab, Darko Kataris, became really interested in this and said, well, what's going on? And so what he did was look at the green ecomorph. And what was interesting here, and this is something I wrote about ages ago, and it has it's somewhat baffling, but you get a very high number of species on this island of East Maui. And this is all of this has stood the test of time, thankfully. But now we're starting to get much more detailed insights as to what's going on. So let's just look and see what Darko did was use what's called exon capture. a genomic approach to really understand how populations of this green ecomorph, you get 3 different species arranged of the same ecomorph arranged across this mountain at different places. So Darko was saying, you know, how come you get so many of these species in this one, on this one volcano? So this was the idea. This was Macrocantha and then Waikimoi and then Brevignatha, the 3 species juxtaposed across with a kind of parapatric distribution across the volcano. So Darko came along and looked at this, and he found that these green, the different species and populations of the green ecomorph were arranged genetically across these volcanoes. What he showed was that he looked at them on all these, on Maui, on Lanai, on the Big Island. For whatever reason, they're not on Molokai. But so he saw very distinct populations. And how best to explain this? So what it seems is that they diverged into different species in these different areas. So one on the Big Island, one on Maui, one on Lanai. So you get this Lanai species, which became Macrocantha. You get the Big Island species, Brevignatha. And then it seems they then went, presumably, there must have been a period of allopatry. And then the most likely scenario is they've gone back to colonize these multiple species occurring on the same volcano. So what was really interesting about Darko's work in particular was that as they came back together, he was able to show that they're not just parapatric, they're actually interdigitated. So you get genetic entities, there's very little evidence of hybridization, but you get these genetic entities interdigitated across the mountain. So this implies that you've got allopatry, genetic isolation, secondary sympatry allows interaction, and then you get subsequent ecological divergence that allows stable co-occurrence. And this is what we're trying to understand right now. One thing that we're looking at is what is the isolating factor? If these things are not hybridizing, what in the world is happening? How do they recognize each other? And this is work by Ashley Adams, who's a current student in my lab. She was looking to see what factors are involved in mate recognition. These things they're nocturnal. It rains a lot in their habitat, and they've got very little courtship. You think of spiders courtship and, you know, waving their legs. These things don't. You put a male and a female in a vial together and just shake them. That's how you get them to mate. They lock their jaws with very little evidence of any courtship at all. So, and then they just, once they've locked their jaws, there's the male on top, the female below, and he's inserting his palp into her epicynum. So it's a very straightforward process. So how do they recognize each other? So what Ashley was able to, she has been able to show is that they differ in chemistry. So she focused on the island of Maui, where you get all of these gobs of species living together. And what she showed was that in different sites, and I'll show you here, she looked at 2 sites, one in higher elevation Waikimoi, that's a Nature Conservancy property, one at lower elevation. So looking at this high elevation site, what she showed, looking at the methyl ethers, these chemical components that you find in the web, and she was able to demonstrate that they select, they can actually pick out the right species based on the smell of the silk. But she was able to extract the chemicals from the silks and show species within, this is lower Waikimoi here, the species differ hugely in principal component space. There's a lot of differences in the methyl ethers, but they separate out such that there's absolutely no overlap at all between these different species that co-occur there. This is in Upper Waikimoi, very close by, but at higher elevation. You get many more species co-occurring at the site, and you see they do not overlap at all in principal component space. The very different composition of their chemistry. So you see how all of this can happen. Basically, what seems to be going on is that you get divergence in allopatry. They come back together. They're isolated. They're they can't mate. And so you get character displacement, both ecological and sexual character displacement, that allows them to recognize each other sexually and allows them to diverge ecologically into different niches. So the bottom line here is that there's a lot of different things going on. And so I've shown, all of these different things in the different lineages. And so you say, well, if there's all of these different things going on, how do we get at generalities? How are we really going to understand the speciation process? And this is why I'm currently working on trying to understand the evolution of entire communities, so community dynamics across the chronosequence. And this would be a whole other talk so I'm not going to get into it, but basically what we're trying to do, and this is work with Henrik Krahenwinkel, who's at Trier University. But Henrik has been, our whole team has been extracting arthropods from different components of the canopy, the understory, the leaf litter layers, and we're getting quantitative samples of arthropods across the sequence, looking at similar habitats and similar sampled in the same way through malaise trapping, through beating, and trying to understand how the entire community of arthropods changes as you go from younger communities to older communities. So what we're trying to understand from that is we're getting at how abundance distributions change across the chronosequence, how diversity patterns change. Do they, what groups diversify and in what ways? We're trying to tease apart these signals to get a general understanding of how things have changed. This also allows us to look at networks, in particular, just basically understanding what organisms are associated with what plants, for example, and again, how these change across the chronosequence. And ultimately, what we really want to understand is what is it about the community that makes it more or less vulnerable to invasion. So we're trying to separate native and non-native and see where the interaction sets in, and how it plays out again across the chronosequence. So with that, I'll just try and summarize what I've said here. So the bottom line is that when you think about adaptive radiation, this is really an umbrella term. The rules of diversification are very different. Even within, I've been focusing almost entirely on the genus Tetragnatha. So even within this radiation, you get very different rules. So first of all, species can multiply by occupying different geographic areas. So this was like the ones occupying the mountaintops and the Orson Wells also, where you get no change in environment or niche. It's so-called non-adaptive radiation. And you saw in the Orson Wells, they just accumulate over time, far the most species on the oldest island. They seem to kind of just go into smaller and smaller areas. So that's one. Then species can multiply by occupying different environments. And we showed this in Tetragnatha by these white ones and the black ones, the paired species that have gone lava onto the lichen, so lava to dry forest, and they've gone from black to white. And so this is you find this in a lot of plants, and as I said, in the wolf spiders, and it's the same kind of pattern that is very well illustrated in stickleback fish. So speciation here is associated with divergent selection into different environments, and you get the formation of kind of ecotypes. Ecotypes generally refers to populations, but here you've got, you know, the formation of different species in these different environments. And again, species accumulate through time. You can get reinforcement through secondary contact because neither of the two species can do very well in that intermediate area. And then the third one is species can multiply through initial isolation within the same environment. And this is what we found that seems to be in the same thing in the anolis lizards, but it's common in the spiny-legged tetragnatha, in some of these web building groups, not all of them. But what happens here is that speciation, as we've shown in the spiny-legged ones, speciation is associated with niche shifts, presumably mediated by interaction with close relatives with which the taxon co-occurs when it comes into secondary contact. Here, they're isolated. As Darko showed in his work, there's no hybridization, no evidence of hybridization. So you get ecological and sexual character displacement as a result of competition when they come into secondary contact. And this is where you get the ecomorphs. You find this also in Ariadne spiders in Hawaii, where you get repeated evolution of the same form. The same kind of thing goes on. The other thing here is that the way that species have of accumulating seems to be entirely different as well. It seems that you get rapid early diversification. These different entities are just seem to be just popping out all over the place. And as you saw again from Darko's work on Maui, multiple entities across this island, this one volcano of Maui. So multiple entities interdigitated. Presumably a lot of them go extinct, but the ones that don't are going to compete like crazy. And then this is where you get divergence, ecological divergence, sexual divergence, and then amongst these, you just get a lot of species on that young island. And then, so they're all competing or going extinct. And then over time, it kind of calms down. And you do not seem to get any increase in numbers on the older islands. So what I want to emphasize here is that the patterns are really quite different in these different groups. And they go along with whole suites of characters. So, you know, you can say, oh, I would expect to find ecomorphs in this situation, but I would not in this other situation. So we're starting to kind of build a picture of what the expectations might be. So this is all very exciting to me, but I want to leave time for just a few questions. So just with that, I just want to thank all the players here. I should say just some of the players. This is Ashley, who's doing all the incredibly pioneering chemical work. Darko, who I highlighted in much of the genomics work. Susan Kennedy, who's done a lot of the behavioral and ecological work on the niches of these spiders. And Natalie Graham, who's doing a lot of the metabarcoding work, and also within the different lineages of spiders at the moment. Of course, all of the people in Hawaii who have been kind of helping to get this going, and I just recognize here in particular Betsy Gagne and Lloyd Loop, both of which Betsy died right at the beginning of COVID, and Lloyd just a few years ago. But they kind of recognize the importance of understanding their diversification of these different groups. And there's Jean-Yves Meyer from French Polynesia. Sam Gone was the one who introduced me to happy face spiders. Art Medeiros, Pat Biley, Ken Kaneshiro. It's that I could go on for another hour. And of course, in the new work that we're doing, Andy Rominger, Junying Lim, and Henrik Krahenwinkel. But that's a whole other talk, as I said. And finally, my husband, George Roderick, who's kind of been helping with all of this throughout.